Okay, so ladies and gentlemen, in today's videos, we are going to look at the lipid transport in the circulation. So after the digestion and the absorption of lipids, the next thing is the transport of the lipids to various tissues. So lipids generally are insoluble in plasma. So in order to be transported, they usually combined with a specific proteins to form lipoproteins. So that is usually happened. Since the lipids they are insoluble in plasma, so therefore it is not possible for them to be transported to the various tissues and organs of the body. So for them to transport it, they need to combine with a proteins to form what we call lipoproteins. So when you have lipids combined with a proteins, so lipids combined with proteins, we generate what we call lipoproteins. That is a combination of lipid and protein. So we generate what we call the lipoproteins. So the proteins are called apoproteins and they are usually attached to the surface of the lipids. And of course, we have cholesterol and then we have non-polar lipids. So generally, when we are talking about these lipoproteins, it's a combination of proteins and the lipids. So generally, the non-protein component, the non-polar component are usually found in the core. They are found in the core, that is inside the lipoproteins. An example of them, we have triacylglycerol and cholesterol esters. So triacylglycerol and cholesterol esters are usually found inside the lipoproteins, while the polar component are found on the surface of the lipoproteins. So this is the only combination that can be transported to the various tissue because it can be solubilized in the plasma. So we have four major classes. Of this lipoprotein. So all of them, they contain characteristic amount of triacylglycerol, cholesterol ester, phospholipids, and lipoproteins, but of different densities. So generally, these lipoproteins, they combine with the proteins, but they produce a different particles with different densities ranging from chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. And of course, we can see that these class of lipoproteins are arranged based on their increasing density. So it means that chylomicrons have higher density, followed by chylomicrons have lower density, followed by very low density lipoproteins, and then low density lipoproteins, and then high density lipoproteins. So it means that they arrange based on the increasing of their density. So it means that the density, because they have different density, of course, due to this different density, that is why you can easily separate them using ultracentrifugation. And of course, they have their diameter. So the diameter of chylomicrons is 500. Very low density lipoproteins have a diameter of 43. Low density lipoproteins have a diameter of 23. And then high density lipoproteins have a density of 8. So it means that the chylomicrons have the higher diameter, followed by the very low density lipoproteins, and of course the HDL, which is high density lipoproteins, have the lowest diameter. And then the next thing is the sources and the function of these lipoproteins. So first, chylomicrons, they are usually found in the intestine, and their main function is to transport dietary TAG, that is to transport triacyl glycerol. So when we take our diet, and if our diet is rich in triacylglycerol, so of course these chylomicrons will be the one to transport it to the various tissues through the blood circulation. And then there are major lipoproteins. There are major apoproteins that are found in the chylomicrons. So these lipoproteins that are found in the chylomicrons, we have apolipoproteins A, alpha lipoproteins B48, and we have alpha lipoproteins C1, C2, and C2, and then finally there is alpha lipoprotein E. And of course, for the very low density lipoproteins, it is found in the liver, and the major function is to transport of indigenously synthesized triacylglycerol. So our body also generally synthesizes triacylglycerol, especially after the absorption. We know that the lipid and fatty acids, the fatty acid and glycerol re-esterify themselves. So when they re-esterify themselves, and of course we have also other methods where there is a de novo synthesis of this triacylglycerol. So the major function of very low density lipoproteins is to transport indigenously synthesized triacylglycerol. And of course, the major lipoproteins for the very low density lipoproteins is B100 and alpha lipoproteins C1, 2, and 3, and then E. So it means that the difference between the chylomicrons and VLDL in terms of the alpha lipoproteins is just that A and B28 is absent in VLDL, and of course B100 is present in VLDL, which is also absent in chylomicrons. And then the next one is low density lipoproteins, and the main function is to deliver cholesterol to peripheral tissues. So it transports cholesterol peripheral tissues, and it only has alpha lipoproteins B100. And then the next one is high density lipoproteins, is found in the liver, and the major functions is actually removes the used cholesterol from the tissues and take it to the liver. And that is why it is even called good cholesterol, because if this cholesterol are transported, like for example for the LDL, is transported to various tissues, like for example artery. So the cholesterol usually clog the surface of the artery, leading to a condition called atherosclerosis. So as a result of that, it may lead to hypertension, which can also lead to so many coronary heart diseases. But the presence of this HDL, if you have it, it usually removed those cholesterol from the tissues like the artery and then take them back to the liver. And of course, the major alpha lipoproteins, we have A, and we have C1, C2, C3, and then alpha lipoproteins D and E. So these are the four major classes of lipoproteins that are found. So then after this, and of course they are arranged based on their according to increasing of their density. So now, what is apolipoproteins? So apolipoproteins, their main functions, one is to provide the structural stability of lipoproteins, and also they serve as a ligand for the interactions with lipoproteins receptors that help determine deposition of individual particles. And of course, these lipoproteins, they have their receptors. So of course, there must be receptors and the ligand interaction. So the alpha lipoproteins are found on the surface of these lipoproteins. They serve as a ligand that usually bind to the receptors for the other downstream application. And another functions of these lipoproteins, it acts as a cofactors for enzymes that involved in the plasma, lipids, and lipoproteins metabolism. So these are the three major functions. First, the stability of the lipoproteins, and second, it serves as a ligand for binding to receptors, And then it also acts as a coenzymes that are involved in the plasma lipids and lipid metabolism. So there are so many types of apolipoproteins. There are so many types of apolipoproteins and how they have their functions. Like for example, we have ApoB100. So ApoB100 is found on VLDL, IDL. When we say IDL, it means intermediate density lipoprotein. And of course, LDL is low density lipoproteins. And the major functions of this ApoB100 is the secretion of VLDL from the liver. So it helps in the secretion of VLDL from the liver. And then the second one, the second function, it has the structural proteins of VLDL, IDL, and HDL. So it serves as a structural protein for these lipoproteins. And then thirdly, the ligand, it serves as a ligand for LDL receptor. And of course, we have ApoB12, which is found in the chylomicrons and the remnant of the chylomicrons. And the main functions is the secretion of chylomicrons from the intestine. So it helps in the secretion of the chylomicrons from intestine. And we have ApoE, which is also found in chylomicrons, VLDL, IDL, and HDL, and also have a ligand for binding of IDL and remnant to low density lipoproteins receptor. There is also ApoA1, which is found on HDL and chylomicrons, and the major function is actually it helps in the structural protein. So it's a major structural proteins of HDL, and it also serves as an activator of LCAT, that is low chain LCAT. So when we said LCAT is an enzyme that is lipids. Yes, it's when we said LCAT, it means low chain. Let me confirm. Yeah. It serves as an activator of low chain acyl transferase enzymes, long chain acyl transferase. And for the ApoA2, the function is not known. And then for ApoC1, the function is also involved in the activation of long chain acyl transferase enzymes. And of course, we have ApoC2, which is also the major function is the activation of lipoprotein lipase enzymes. And of course, we have ApoC3, and the major functions of this ApoC3 is actually serve as an inhibitor of lipoprotein lipase activity. So C2 is activator, and then C3 is inhibitor. So now, the plasma lipoprotein structure, how their structure look like. So we have the lipoprotein core, which is inside the lipoproteins, and it's made up of cholesterol esters and triacylglycerol. And of course, we can clearly see from here, that in the core we have triglyceride, and then we have cholesterol esters. Then after this, we also have the surface of the lipoproteins is made of phospholipid proteins and cholesterol, and of course, that is what makes it soluble lipoproteins, because of the nature of the biomolecules or the structure of the compound that found on the surface, which comprises phospholipids, proteins, and cholesterol. And of course, from their structure, they have a polar combine. So that is why they interact with the plasma for the transportation of the lipids to various parts of the body. So this is actually the composition and the properties of human lipoproteins. Human lipoproteins. So in this case, we said we have the lipoprotein class HDL, LDL, IDL, VLDL, and chylomicrons. So this is their density, their normal density in humans. And if they are above that or below that, it means that there is a problem. And of course, this is the range of their diameter, and then their protein or percentage of their protein of dry weight. And of course, the HDL is the one that have the highest protein percentage. It has the highest protein. So actually, we can be asked to actually classify or arrange these lipoproteins according to their protein percentage of dry weight. And of course, you should know that the HDL is the one that have the highest percentage of protein in it, and then the chylomicrons is the one that have the least proteins. And of course, in terms of the phospholipids, the phospholipids in the case of HDL also have the highest percentage of phospholipid, and then the chylomicrons have the lowest percentage of phospholipid. But reversing the case is the case in terms of the percentage of triacyl glycerol of the dry weights, which is the HDL have the lowest triacyl glycerol content, while chylomicrons have the highest percentage of the of the triacyl glycerol. So it means that chylomicrons is the one that accommodate most of the triacyl glycerol. So you should be able to know this and identify which one have the highest protein, which one have the highest phospholipids, as well as the triacyl glycerol. So the composition, like for example, most protein have a densities of about 1.3 to 1.4 gram per ml, and the lipid aggregate usually have densities of about 0.8 gram per ml. So now the next thing after this is let's look at the different plasma protein classes and their function. So chylomicrons is synthesized in the intestine and it transports dietary lipids, and of course 98% of, in terms of its chemical composition, 98% of the chylomicrons contain lipids, and it have the large size. It is the one that have the largest size as well as the lowest density. And of course, it has APO, B48, which serve as a receptor binding, APOC2, which serve as lipoprotein lipase activator, then APOE, which serve as a remnant receptor binding. And then the next one is very low density lipoproteins is synthesized in the liver and transports the indigenous triacyl glycerol, and of course 90% of it is made up of lipid, and 10%, the remaining 10% is for the protein, and it's made up of ApoB10, which have a function of receptor binding, ApoC2, which serve as a lipoproteins activator, and there is also ApoE, which serve as a remnant receptor binding. And this is the IDL, is synthesized from VLDL during VLDL degradation, and the main function is transport. It transports triacyl glycerol and precursor to LDL, and it has ApoB100, which are for also receptor binding, and then ApoC2, which serve to serve as an activator of lipoprotein lipase. Then we have ApoE. And then we have low density lipoproteins. It's synthesized from IDL, and the main function is to transport cholesterol, and 78% of it is made up of the lipids, and then 58% cholesterol. And then it has ApoB100, which serve as which also have the functions of receptor binding. So this is the LDL In terms of the structure, it have contain cholesterol, phospholipids, unesterified cholesterol, and then lipoproteins B2. So, ladies and gentlemen, this is where we are going to stop for the transport of the lipids. And if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.